now to introduce our presenter for tonight's lecture, Kate Goodrich. Kate Goodrich is a professor of biology at Widener University in Chester, Pennsylvania. She earned a bachelor's degree in theater prior to entering graduate school for botany, where she earned a Ph.D. in plant biology in 2008 from the University of South Carolina. Her doctoral research focused on the floral scent, color, and architecture for all species in the genus Asimina, pawpaw and close relatives. Kate came to Widener University in 2009, and over the past 13 years, she has worked with numerous undergraduate student researchers in her lab and at field sites. Her research has repeatedly returned to Asimina triloba, common pawpaw, over the years, including a biomechanical investigation of the twisty twigs in pawpaw. More recently, Kate has been studying floral mimicry, which brought her back to studying floral features of pawpaw, and an investigation into the pollination strategy for pawpaw. This study spanned 5 years and was recently published in the International Journal of Plant Sciences. I can't wait to learn more from Kate tonight, so at this point, I will turn things over to her to get us started. Welcome, Kate.
Thank you, Amy. Thank you, everybody. I can't see you, but thank you for being here. I'm excited to talk about pawpaws. It's what I do a lot of, so I'm happy to share this. So like Amy said, I am going to be presenting tonight a little bit about the pollination of this wonderful species. Right now, you hear more about pawpaw in September because it's making these fantastic fruits. And if you've ever had the fruit of pawpaw, you know what I'm talking about. If you haven't, then you might be in luck because we're sort of in that window of time right now where you start to get fruit dropping from the tree. And I'll tell you, this is a pretty short window. So if you can get the fruit, get them now. All right. But we're going to go back a little bit in time, and we're going to talk about how you get to those fruit by talking about how the flowers get pollinated. And to do that, I am a professor at Widener University, and part of what I do is teach, so I can't help myself, but I'm going to teach a little bit as we go through this. I'm just going to go through a little bit of background on pollination in general to catch everybody up. So pollination is broadly what I like to study, and that's, usually involves insects or other animals that interact with flowers. This is a textbook example of a flower. It is not a pawpaw flower, so I apologize for that. But it's a nice textbook example. It's a lily of some sort. And just as a quick reminder, you've got male parts of the flowers. I'm hoping that people can see my pointer. But there's a stamen over here. That's the male part. It makes pollen, which is going to help to fertilize the eggs, which are down here. This is the carpel, the center part of the flower. It's the female part. And when pollination happens, you've got to move the pollen grains from the male part to the female part. Now, a lot of plants have what we call bisexual flowers, so they're going to have both parts, but they don't really like to pollinate themselves a lot of times. So what that usually means is that you get cross-pollination going. And if anybody gardens, then you already know this pretty well, probably, but flowers usually attract some form of pollinator, insect. In this case, we've got a bee. And even if a plant has both male and female parts, it attracts in insects usually to move the pollen. And either the structure or the function of the flower is such that the insect will pick up the pollen in one flower and then travel into, some amount of space to another plant of the same species, enter the next flower and drop off some of the pollen as it goes into the flower at the female part. And that achieves what we call cross-pollination. So it's not done once the pollen moves. Once the pollen gets moved to the female part, the pollen actually germinates and grows down into the ovary, and that's where fertilization is going to happen. So that's where you get seeds formed. Seeds are going to be little tiny embryonic plants, and again, a lot of you probably garden, so seeds go in the ground and the seed germinates and the new plant grows. So pawpaw fruits, if you are a fan, have big black seeds or dark brown seeds that are about this size, about the size of a quarter, a little smaller. And each fruit contains a number of seeds. So this is, a carpel that will have a number of different egg cells down there that get fertilized. All right. So again, if you're interested and excited about the fruit now, you got to start out with the flower. And the flower actually happens, you can see my cursor, back in about April or May of the same year. Okay, so this past spring, there were a bunch of flowers on the trees. And if we were lucky and they had some really helpful pollinators there, then you end up with these big potato-looking fruits. To me, they always look like green potatoes hanging there. They're kind of ridiculously large given the size of the flower that it started from. So, all right. So pollinators in general are a wide range of different potential animals. Most of the time they're insects. Of course, there are bats and hummingbirds and things like that that pollinate as well. But I just want to remind everyone, we think of bees as being great pollinators, and they are, but you also have beetles and butterflies and flies, all sorts of things that can pollinate. So I'm interested in this relationship between flowers and insects. And in particular, I'm really interested in the smells, the chemistry that come out of flowers that attract the pollinators. And within the broad scope of pollination biology, I'm interested in the kind of pollination that involves a flower deceiving a pollinator by pretending to be something it's not. I find this just really fascinating on both an ecological scale and an evolutionary scale. So we call this floral mimicry. This is a flower pretending to be something it's not. The flower we refer to as a mimic, and whatever it's pretending to be, we call the model. In a few cases, it might be a flower pretending to be a different species of flower, so it can pretend to be somebody else. In some weird examples, you've got flowers, you may have heard of this, but orchids that look a lot like a female insect, and they get pollinated by the male insect, and they actually put out a scent that smells just like the female insect, and the male tries to mate, literally tries to mate with the flower, and it pollinates while it's doing that. So that's another kind of mimicry that's really fascinating. The kind of mimicry I study is neither of those. I'll get to that in just a second. But the idea is that once you have successfully fooled the insect, it should pick up pollen in the process of interacting with whatever it thinks you are. And you need to fool it more than once because it's got to visit you, but then it's got to carry your pollen to another flower of the same kind and be fooled a second time. And if it does that, then you achieve pollination. So in order to be successfully pollinated when you're a flower that is practicing floral mimicry, you've got to be really convincing. Okay, so I find this really cool because obviously there's a lot of cool ecology that goes into it, the ecology of the insect and what it's expecting to find, the ecology of the flower that's tempting in this insect that's looking for something non-floral. Evolutionarily too, this is really fascinating. This is just a sort of simplistic diagram to give you an idea in terms of evolution of the flower, how a flower gets to look like something that it's not. Where initially you would expect it just to have perhaps a few cues, like maybe a certain smell or a certain similarity visually to something that attracts an odd insect that makes a really good pollinator. And this is the fooled insect that's being duped into visiting. And over evolutionary time, if you're following this, evolution of the flower, you can find, ways that its phenotype, its scent, its color, its shape, different aspects of the way it's displayed, how that becomes increasingly a match, what we call a multimodal match, to whatever that model is that it's pretending to be. And that should increase its fitness. And by increased fitness, that basically means it makes more seeds, it's more successful, it makes more copies of itself. So it's a cool way to study evolution too. And both these ways, I really enjoy studying floral mimicry. Okay, so the kind that I study, these aren't sexual mimics where they pretend to be another insect that the insect wants to mate with. They're not the kind of mimic where a flower pretends to be a different kind of flower that might be more rewarding or something. I study flowers that specifically pretend to be something that is dead and decomposing. And I know that sounds a little gross, but to me, it's really interesting how this works. So forgive the imagery here, but you have flowers that are pretty convincing mimics of dead animals, of rotten fruit, of other fermenting substrates that might be in the environment, and even poop. So there's a whole bunch of different kinds of mimicry in this subgenre of floral mimicry. And what I really like to study is how the floral phenotype is composed, like what smells is it putting out? And you can imagine these are pretty stinky smells. Or, what is the texture of the flower and what color is it and what kind of insects does it attract? So when a flower pretends to be one of these decomposing substrates, it naturally attracts a bunch of flies that you might associate with these things. And these aren't the kind of flies that you normally associate with flowers. There are certain kinds of flies like syrphids and bombylids, these little hairy striped flies that look a lot like bees that honestly like flowers. They are adapted to floral resources and they're looking for nectar and pollen and things like that to eat. The flies that are involved in these systems that I study are not those kind of flies. These are not flower adapted flies. These are the flies that very much want to go to something that is gross and dead and stinky, and they feed there, they find mates there, and they lay eggs there. Okay, we're getting to pawpaw, don't worry, but first I'm going to just give a few quick examples of other plants that do this you might be familiar with. This is a Stapelia, I think grandiflora, but it's a relative of the milkweeds. And this particular group grows, I think, in Southern Africa, South Africa specifically. And the flowers are pretty amazing. They look like a giant starfish. They grow close to the ground. They're this sort of mottled purple yellow color. They're covered with hair. So this isn't hair like animal hair, but it's a flower's attempt to look hairy. And they put out this powerful stench that smells like a dead animal. And in doing that, you attract flies. And this is a great story with this picture because it's from a friend of mine in South Carolina, which is pretty far outside the natural range of this plant. But even on a whole different continent across the ocean, this plant still is successfully attracting flies. And those flies are so convinced that this is a dead little animal that they've laid their eggs in it. And that's a huge cost for the fly because now its babies will hatch and they won't have anything to eat because it's not a dead animal, it's just a flower. So it's really cool the degree to which some plants go to this mimicry. This is just one other visual example that I think is pretty striking. This is a species of Rhizanthes growing in, I think, Borneo. I didn't take the picture, a friend of mine did and shared it with me. But this little flower is growing on the rainforest floor, and you can see it looks just downright furry. It looks like a pelt. And the tips of the petals are oozing some sort of grossness, and it looks like it has a tail attached to each petal. I mean, it's a very convincing visual tactile mimic. And on top of that, this flower also puts out a really powerful stink. It attracts flies, and you can see a fly sitting there. And again, it's doing the same thing. It's attracting in flies and hoping that they will move pollen from it to another plant with the same flower. So crazy. All right. If you're worried that you've just bought a pawpaw tree or something and now you're going to have the smell of rotten flesh in your yard, don't worry. This is another mimic, likely, but it doesn't smell like a dead animal. It smells more like the fermenting fruit that I mentioned earlier. So these flowers are cute little, about 3 centimeters, 2 centimeters across, maroon cups. They smell like baking bread, really. They smell very yeasty. They don't really smell fruit-like, honestly. They just smell like straight up yeast fermentation. A little bit like beer or wine, if you know that smell. It's a very pleasant odor, I think. Not bad at all. So these small little flowers come out in mid-April till mid-June, or sorry, mid-May. And they put out a whole bunch of scent because the tree will be covered in them. If they get pollinated, put out some of the largest fruit in North America, these wonderful pawpaw fruit. And they have a whole history in the North American continent, largely the eastern half of North America here, with indigenous cultures. So they were certainly well known long before colonists came to this country, and were a huge part and still are a huge part of indigenous culture and cuisine and all of that. So they have a really interesting cultural component as well as evolutionary and history and ecological significance. All right, so here are the flowers of pawpaw close up and just a little bit of biology about them. They are bisexual flowers, meaning that they have the male and female parts. And they separate those so that they can cross-pollinate, get their pollen to other plants by separating the sexual phase in time. So when they first open, they're female. This one's the female stage one here. You can see the carpels are nice and receptive, ready for pollen to stick to the tip if a fly comes in or some insect comes in covered with pollen. A few days later, they progress towards male stage, and at that point, they're probably no longer receptive, so hopefully those carpels got fertilized and fruits are starting to form. And at that point, this tight ball of anthers that you can see here all closed up and packed together, will open up, and they release a lot of this really large grain pollen. It's really sticky, and it's nicely visible if you're looking for it. All right, and as I said, if you're lucky, a couple of carpels, maybe 3, even 4. I've even seen clusters of like 5 or 6 in really amazingly large groups. Those fruits will form one from each carpel in the flower. So if the flower has multiple carpels, you'll get this cluster of fruit hanging off of a single stem. So it's pretty impressive that one flower can make all of that. Okay, so the research that I've done over the past 5 years or so with some students at Widener in my lab asked 2 questions. The first one is, who pollinates? There's a lot of information online and with people that grow pawpaws in orchards and things like that about pollination, but there were no formal studies that documented a huge number of pollinators. There was one done in 1980, but they only found about 60 insects overall, and they themselves said that they couldn't draw a very strong conclusion. So I wanted to find out who the pollinators were in our area of Pennsylvania. And then I also wanted to ask, is this mimicry, since I'm so interested in mimicry? The first thing I wanted to know is what possible models might there be in the environment around the pawpaws in early spring? How well does the flower imitate the model? So how much overlap is there, especially in the scent chemistry, because that's really what I'm into. And then finally, if we've identified who the pollinators are in the first question, we want to see if the same insects are active with the models and interacting with those to help solidify that that's the actual model. And before I move into some of the details of that, I just want to introduce these 3 young women who were all fantastic. They're all Widener students who worked with me for multiple years in my lab, what they've done. But they also are all co-authors now in the paper that we just got out from all this work that we did. So they're fantastic. Okay. So I'm hoping that everybody can hear me because my computer says that I'm a little unstable from the internet. So just, Amy, let me know if I freeze. This is a slide just showing you 5 years worth of data of all the insects that we recorded visiting flowers. So that's a very long time to watch these flowers. And if you go out and you watch flowers in the springtime from pawpaw to see pollinators, there's a reason that the study wasn't done earlier or just in a year's time. The visitors are very few and far between. It takes a long time to get a good count of insects going to the flowers. Some days we would sit out there for an hour or 2 and you'd see absolutely nothing. It's just amazingly tedious sometimes. And then other days, and I can't even tell you what the perfect conditions were. Some days that were really active were rainy days that were kind of windy. But anyway, for whatever reason, some days you would go out and there would just be insects moving all over. It was really exciting after days of nothing. So with a lot of patience and about 5 years worth of time, we collected a pretty good set of observations. You'll notice that the most numerous insects there are Drosophila, which are little fruit flies. These are tiny little flies that are about a millimeter long. But there's a decent number, more than a third out there were larger flies, medium large flies, and then a very, very small number of other insects. Beetles, I know, are occasionally associated with pawpaw flowers, and we did collect one beetle, but from the overall observational study, we only saw 2 butterflies that were visiting and probing for nectar, and I think 3 vespids, like little wasps, that were in there. So really, flies are the story here. Okay, so I've got some pictures of flies. This is just to kind of drive home the part about who pollinates, because sometimes the most frequent visitors are not necessarily the most effective pollinators. So the first pictures I'm going to show you are flies hanging out in the flower with just an absolutely ridiculous amount of pollen on them. The one over here that I used to advertise the talk just has a little bit of pollen. I'm trying to get my pointer up to show. But over here, you can see this fly is just coated. It has a thick coat of pollen on its back. That's really, to me, pretty amazing. Same thing with these. You can see this one over here. Just, I mean, look at all of that pollen. That's just crazy. And no, I did not dip a fly in glue and glue it to the flower and stick pollen on it. Like this, it just blows my mind that that's, you know, a real fly, that much pollen. All right, more flies, more pollen, lots of pollen. So you'll notice all the flies in these pictures are the size of house flies, right? Muscidae, Californidae, these flies that are, you know, pretty robust, medium to large. Now, if you remember the pie graph that I showed you, Drosophila, the fruit flies, are the most numerous visitors. And those are the ones I've circled here, but based on the flower's shape and where the sex organs are and where the pollen is, these flies rarely come into contact with that. And I have no doubt that they probably contribute a little bit to pollination because they crawl all over inside the flowers. And at some point, I'm sure they pick up several grains and happen to move them. But if you're talking about effective pollinators, I would totally put my money on the medium to large flies with those pollen loads that they're carrying on their back. Okay, so I think we got pretty good results for the who pollinates piece. The next question was, is this mimicry? So we had to go out and search for potential models in the environment. We compared the phenotype of the model and the flowers, specifically floral scent and the scent of the models, to see if there was a lot of chemistry overlap. And then we watched as many models as we could for as long as we could and collected insects off of those to see if the same insects that were in the flowers also visit the models. So here's some of the best bets we have as models in the environment. Before I started this study, I didn't know what a sap flow was, but sap flows are actually, turns out, pretty common. If you've looked closely at the bark of mature trees, especially in the springtime, sometimes you'll see sap leaking out through maybe some damage in the bark, or it could just be a crack in the bark for what have you reason. But in springtime, if you know what trees are doing, they've stored a lot of sugar. Well, okay, this is in a temperate area like where we are up here in Pennsylvania. A lot of sugar gets stored underground in the roots. And in the spring, when the temperature starts warming up, you'll have a huge flux of sugar and sap going up the tree trunk out to the branches to help do bud break, to grow all those leaves that make the canopy of the tree that you need for getting the tree's food in the spring and summer. So springtime, when pawpaw is blooming, is a great time to also have sap flow going up the trees. And if there are breaks, like I said, then you might get these sap flows out of the tree. And yeast is pretty ubiquitously everywhere in the environment. So as soon as there's sugar leaking out of the trunk of a tree, you're likely to get fermentation happening. So I think this is a pretty plausible idea of where fermentation-loving insects, especially flies, are going to be hanging out in the early spring if they get tempted away to a pawpaw flower, right? There aren't a lot of ripe fruits in early spring. It obviously takes a while for flowers to bloom and then make fruits and all of that. So fruits seem like a good model, but there just really aren't a lot around in April and May. Ripe mulberries were the very first fleshy sweet fruit that we found at our field site, and that happened usually about 2 weeks after blooming time, but it's still relatively close to the time the flowers bloom. So these drop to the ground. If you've ever tasted mulberries, they don't have a strong fruity flavor. They're just very sweet, but they make a great source for fermentation. So there was a nice hefty smell of fermentation when we got close to the mulberries on the ground. And then luck would have it, if you want to consider it luck. From my point of view, I do. But obviously something like a raccoon was eating a lot of these mulberries, because then we found dung nearby that had a ton of mulberry seeds in them. So obviously this animal had passed a lot of mulberries. And in terms of the sort of plants pretending to be decomposing substrates, dung or other, feces come up a lot as a potential substrate. And so one of my questions here is, with yeasty things, if an animal eats a lot of yeasty fermenting fruits, is the stuff that comes out the other side also possibly a substrate? So hence we collected this. Okay, so I wanted to give you a little bit about how I do the scent stuff in case you're interested. I hope people are, but I've done this since my graduate degree, so I've done this for a long time. The method that I do involves collecting scent on something that's sort of like a pen. This thing here that's in the red circle, the red oval, is about the size of a big pen, and it's got a fiber in it to which scent will stick. So it's a way of collecting scent by having it stick to something solid. And if I'm collecting stuff out in the field, like mulberries or dung or something like that, I can put something like this plastic bucket over it, and it collects the smell inside the airspace that's contained. Then I put this fiber down into the airspace for an amount of time, let the scent stick to the fiber, and then I take it back to the lab. So it's really not that complicated, and it's actually really great with undergraduate research students because it's easy to learn and they're real quick to pick it up. Okay, so the quick details on how you sample this: you take that fiber, you inject it into this machine with a really hot oven. So when the fiber goes in, it heats up to a really high temperature, and that liberates any of the scent molecules that are stuck to it. All of them get fed onto this very long column. And if you remember, maybe from high school or college chemistry class, chromatography is a process by which you separate a mixture of chemicals into individual components. So what this does is it takes the scent sample from the flower or the poop or the mulberries or the sap flow, and it takes the mixture, separates it out into its individual ingredients, and then at the end of the column, let's get my, there it is. Well, it's sort of white on white. So grab a pointer so I can actually tell you. Okay, so each compound comes off the end here and gets broken up into little pieces. And it's that breaking pattern. So there it separates. When it breaks apart, that's what tells me what compound it is. There's a library in the computer that can help me match it. And I've just been doing this for years, so I've learned a lot of the patterns. So anyway, it's that breakup pattern that tells me what the chemical is. So I do a lot of this scent research. Just to give you an idea of what a pawpaw flower smells like, this is the data from that. And you can see each peak represents one of those individual compounds coming out of the machine, and it detecting that something is coming out of it. And then the fracture pattern that goes with it is down here in the little boxes. So for peak number 1, this is its fracture pattern, and so on and so forth. There's 2 arrows for peak number 5 because there's 2 different isomers of the same compound, and they both look like this when they fracture. So this is the major 5 components. There are a lot of little ones, but the 5 major ones are these that make up pawpaw floral scent. And these also happen to be the same chemicals that come out of yeast if you add them to a little bit of sugar and water and maybe a little flour. So it really is a yeasty smell. This is a big data table and not always the most fun thing to show to an audience, but I just want to give you some idea of, this is the list of chemicals, kind of like from the previous slide, that's in pawpaw. These are the pawpaw floral scents, and this is just a heat map. So the darker the color is in the little square, the more intense that particular chemical is in that sample. So this gives you an idea of the ratios and all for the flower scent of pawpaw flowers. And next to that, you can compare the chemistry from a couple of dung samples. This is with mulberries in the dung, the mulberry samples themselves, some bait that we made up to try to trap flies if they were interested in fermented stuff. It was basically like pancake batter with some yeast in it. These petals are actually petals of magnolia that had bloomed and then dropped to the ground and were decomposing. And then here are some of the sap flows. So what you should take from this is that a lot of these different substrates that I pulled out and I'm showing you here have the same chemicals in them, but at different ratios. And you know, it's kind of hard to know what matters to an insect, to a fly, but it certainly does show that there's a lot of chemical overlap, and that's helpful. All right, one other way to look at really complex chemical data is through multivariate analysis. And it sounds complicated perhaps, but it visually is a lot more intuitive to understand. So in this particular thing, what I've done is I've fed in the entire scent data set of all the samples and all the chemicals and let it analyze sample by sample comparisons. And where they come out on this 2-dimensional map is a representation of how similar they are for the entirety of their data set, the entirety of all the chemicals in them. So you can see all the flowers are pretty close together because they share a lot of the same chemicals and the same ratios. And if you look out from that, it can give you an idea of what some of the most similar things were. And the mulberry samples are pretty close. The dung samples also pretty close. This is some of the bait that I put out. These are the magnolia petals, so on and so forth. So you can kind of see spatially how similar or different the different things are that we compared for models. Okay, I'm sorry, another table, but it's good information here. So you remember the little flies, the little fruit flies were not so great as pollinators, probably because of their morphology. They don't quite fit the flower in order to get pollen all over themselves, but there were a lot of them. And the nice thing from this bit of data is that the most common fruit flies here at the flower were also commonly found in high numbers in some cases on some of the different substrates. And what that tells me is that the flies that have normal ecology of visiting these kind of substrates to feed or to lay eggs are also going to our flowers. So that's useful in looking at this as mimicry. And then the other flies outside of this list are part of those medium to big size flies that are quite good at carrying pollen. And the only thing that I really want you to notice from that is that all of these bigger flies are really diverse. It's not a whole lot of just one or two kinds of flies. It's a whole bunch of different species that are in a whole bunch of different families. So to me, this is really interesting in terms of this whole big community of flies that are fermentation loving. A lot of them, you know, you wouldn't expect to see at a flower. And here they are in pawpaw flowers. And we did actually catch one beetle that we were able to identify. So there was one beetle in a flower that we collected. Oh, and I just want to mention to you that David Grimaldi is, he's an expert at Drosophila, so I give him credit for identifying all of these. And there were a whole bunch of individual flies that he had to identify to species. He's at the American Museum of Natural History in New York City, so we really thank him for that. And Dr. Jade Savage, she is a colleague of mine in Canada at Bishop's University. She did all the rest of the flies, so she did a huge amount of work for this project. Okay, so question, is this mimicry? I'm just going to get the laser pointer, not so obvious. Yes, at least so far we have shown that that the flowers and the substrates that we looked at share a lot of chemistry. There's a good overlap in the chemicals that are put out from both. The most common insects that go to the flowers are also insects that we found at the different substrates. So that was nice confirmation that these are flies with an ecology that brings them to feed on and perhaps lay eggs in fermenting substrates. And overall, yes, this is pretty well supported by the evidence that we had. It was nicely supported enough to get this published. So we did publish it finally this summer. In June, it came out in International Journal of Plant Sciences. I'm very proud to have 3 undergraduate co-authors on there with me. They're very excited about this too, as well as Dr. Savage from Canada. And now for just a little bit of fun stuff about pawpaws in general. If you successfully go through all of the pollination in the spring, then hopefully you end up with a bunch of fruit in the fall. And if you remember that map that I showed you of the eastern United States, is basically covered with pawpaw. So there are a whole bunch of pawpaw festivals that happen basically on this side of the Mississippi and even a little bit west of the Mississippi, celebrating this fruit in the September, October timeframe. The biggest festival that I know of is in Lake Snowden, Ohio. So if you ever get the chance to go, you should go. Unfortunately, pawpaws bloom right when the semester starts. So I'm always in class and I haven't been able to get out there yet to visit it, but someday, maybe when I retire. I also love their posters. I think these are fantastic art posters. So they're really cool. If you want to learn more about pawpaw in general, some interesting natural history aspects, as well as some of the cultural significance of pawpaw, I highly recommend Andy Moore's book. He, I think, published this in 2012 or 2013, but I met him while I was doing some work with another one of our collaborators. And anyway, he's really knowledgeable and it's a fun read. It's easy to read. The other person that Andy and I were with when I met Andy was Neil Peterson. And Neil I have known for quite a while. He is in West Virginia. He has a whole orchard of pawpaws down there that he maintains. And if you visit his website, petersonspawpaws.com, there's a tremendous amount of information about the fruit and the flowers. But he's also a great resource if you wanted to buy certain cultivars to get specific fruit quality. He's your person. So Neil is fantastic. Here are a couple of different articles that I pulled out that are of interest. The 2011 article from NPR actually features an interview with Neil, so you learn more about him and hear from him in person. And then the West Virginia Public Broadcasting segment is from 2020, and it's got some really nice coverage of pawpaw indigenous roots. So if you're interested more in that part of the culture of pawpaw, check that one out. I'm hoping I saw this advertisement a couple weeks ago, but if you're looking locally for pawpaw, if you know where Wolf's is, it's near Media. They were just advertising on Facebook that they have a bunch of pawpaws in season right now. So check that out. This link here, I think is from Arkansas, so it's not a local link, but it's got a whole bunch of pawpaw fruit recipes. And I highly recommend that too. I will say the only thing that I've ever really made with pawpaw consistently is basically banana bread, but with pawpaws. So give that a try. It's fantastic. And that's it. Thank you all very much for listening to the talk. Of course, want to thank a bunch of people for all this work that we've been able to do for the past few years. I want to thank Jenkins Arboretum and Amy for inviting me to give the talk. I had fun preparing it. I had fun giving it. And I thank you, the audience, for listening to it. So with that, I will take questions.
Thank
And
you so much, Kate.
No problem. You want me to stop sharing? Yes.
Yeah, that would be great. And then we can be side by side again. All right.
Yes.
So I'm just going to read the questions to you.
Okay.
So the first question that came in talks a little bit about growing pawpaws. So you can be honest if it's not your forte, but is fertilization better if the tree is in an open area versus like in a woodland?
So it's not my forte. Neil would be great at answering that question, and you can find his email on petersonspawpaws.com. I will say, pawpaw in its natural habitat really likes shade. So I'm not sure about the fertilization aspect, but in terms of it doing well, it will thrive, pretty well in shade. It does well at edges. So we've got a couple of populations that I use for the study that are right at the edge of the forest, and you end up with really full branches, nice, the foliage is super dense. And I don't know about full sun. I haven't seen a lot growing just out in sun. Sorry, I'm not really answering your fertilization question.
No, you're totally fine. This question is actually a question I had as well, which was kind of like if you are growing pawpaws or have them kind of in your landscape or in a garden setting, is there anything you can do to kind of attract those pollinators to your garden? And so this person was asking, should we put out yeast or mulberries near the pawpaw trees? Have you ever thought about that piece, attracting the pollinators?
Well, so I will say flies are frustrating. They're so frustrating because when you don't want them, they're there. And as soon as you actually really want to see them around, yeah. So we didn't actually try to spike the number of pollinators that were coming, but we did put out baits because we had trouble finding natural fermentation sources that were right near the trees, and we were just limited in human power. There were only a few of us out there at a time, so we couldn't be staffing all the possible models. So what we did was we put bait under the trees to try to catch the same flies, and it certainly was successful at bringing in a bunch of flies that were also visiting the flowers. I'm not sure that it increased the number of flies to go to the flowers. I've read a bunch of horticultural accounts of pawpaw, and I know some people were putting back in the 60s, Robert Crowell described the whole genus Asimina, and he basically likened pawpaw to smelling like decaying meat. He said it was fetid. And so then people were putting meat out under the pawpaws to attract flies. And well, they don't smell like meat, but some of the same flies that go to meat will also go to fermentation. So long story short, there's a lot of anecdotal stuff. I don't feel like flies are reliable enough to suddenly be more populous if you put cans of fermenting stuff under them. It might work.
Sure.
But it's a great question. I don't know.
Yeah. This one speaks to a little bit about your final slides with the Pawpaw festivals. Someone apparently knows that there's a Pawpaw festival outside of York, Pennsylvania tomorrow. So if anyone doesn't have anything planned tomorrow and you're looking for a Pawpaw festival, you could head out to York. So thanks for sharing that information.
A link to that, throw it out in the chat or something. That might be fun.
This person's question has to go back to kind of the floral structure. It says trioecious. Is that the correct term for pawpaw? How common is this sexual system in the plant world?
Dioecious, is that maybe? So for dioecious versus monoecious, you're looking at it means house, and dioecious is where you have two houses, meaning that you have a sex that's female, like all female trees, and one that's all male. Pawpaw is monoecious, so it's all together in one house where you have the male and the female there, if that makes sense. It's dichogamous, meaning that the female and the male function happens at different times. The funny thing there is that it would make sense to do that with the tree being like, say, entirely female at once, and then having a tree nearby be entirely male. That would assure cross-pollination. Pawpaw separates in time, but it'll have like two flowers at the same node, right, coming out of the same spot, and one will be male and one will be female. So even though it separates in time, it's not real good at keeping its own pollen out of other flowers on the same tree, which is one of the reasons that they think that the fruit set is kind of historically pretty low, because it doesn't really like to pollinate itself.
Very interesting. This question is, how did you first get interested in pawpaws?
I mean, that's a long time ago because I did my dissertation on the genus and I'm old. But I started in like, I think it was 2001. I was working with a woman up in New Hampshire and she was just a botanist. I was working in a hospital and I thought botany was cool. And she wanted to do scent work. And she actually studied stuff that was in Madagascar, like way far away, and to teach us the techniques that she was going to use in Madagascar. And I was possibly going to go with her. We went down to South Carolina and studied pawpaws because pawpaws come from a tropical family. And so I don't know, I guess the thinking was that the tropical nature of this particular genus might help us. I don't know. Anyway, I got this huge data set as this very naive undergraduate. Well, I graduated from undergrad, but I wasn't in grad school yet. And the guy who gave it to me also ran a lab. And when I did the whole analysis for it, that got me into grad school. So I guess pawpaw is the reason I went to grad school. But the fact that I landed on it just kind of happened.
Yeah.
It was a good choice. Good career choice.
This question is about the observation process for looking at the pollinators. It says, can you automate the initial observation process by using video or still photography?
Some people do that kind of thing for pollination. And the frustrating thing is a lot of times if you're training a camera on something to observe pollinators, like this happens in the rainforest and stuff where it's like a really big flower or a very rare flower where you have one target. Pawpaw is just loaded with flowers. And so sort of the strategy of filming and then counting visits within a degree of focus of a particular flower would be a little less effectual than standing there and being able to, with human eyes, see a whole bunch. I mean, it certainly would save time, but it also then would require multiple cameras and money that I don't have. So it's not a bad idea at all. And again, it works great if you're studying like a rare flower, like an orchid in the rainforest or something where you're sort of training on one particular spot. But I just employ undergrads.
This question goes back to that kind of male-female flower and when they're both receptive on the tree. It says, can flies on male flowers pollinate female flowers on the same tree?
Oh, yeah. Yeah. I mentioned briefly a study in the 1980s. It actually came out in 1980 with Doug Shumsky and Mary Wilson as authors. And they did research on pollination in pawpaw in Illinois. They went out for 2 years and they got extremely frustrated they were trying to measure pollinators. And I mentioned they only saw about 60 insects over that 2-year period. So they had a very low sample size. And then they tagged, I forget, it was something like 12,000 flowers. It was a huge number of flowers that they sort of kept track of. And out of that, it was 0.5% fruit set. So, 0.5% of, there was not a lot of fruit. So to that end, I think there's a lot of pollination that happens that's within the same tree. The tree has dozens, hundreds of flowers on it. And the plant just generally doesn't like to pollinate itself. So it's got a very, very low fruit set from that. There are cultivars that fruit like crazy. So if you get, again, a specific cultivar that's known for its fruiting capacity, and Amy, you mentioned at Jenkins that there's some really high fruiting trees, you can get lucky. So if anybody's growing pawpaw, don't lose hope. It does help if you have 2 trees that are not genetically identical near each other. And then some trees are much better at fruiting than others. So.
This speaks a little bit to maybe planting companions. It says, I heard that it's good to plant native ginger under pawpaws to attract the pollinators for pawpaw. Is this true?
Maybe. I mean, so I'm assuming native ginger is like Asarum canadensis, little low growing.
Yeah.
And the flowers I think are out at about the same time, but I'm not real sure there's strong pollinator work on that, in the sense it's little brown flowers. They're beautiful. I love those. I know some of the species are pollinated by fungus gnats. I don't know of a pollination study that has shown it's the same fly pollinators, but I will say they are beautiful ground cover. I love the leaves that those plants make. So by all means, please plant Asarum canadensis under pawpaws. I think it will look lovely if it doesn't attract more pollinators.
As I'm scrolling through the questions, there was an exact same question about planting native ginger under pawpaw trees. So it is a question that's out there.
The flowers look a bit alike. They have these three petal kind of shape and dark brown, maroon color.
Yeah.
I've gotten down on hands and knees and sniffed them, and they don't have the same odor. I don't really detect an odor from wild ginger.
Right. I would agree.
I have not sampled it either, so.
Yeah. So you did mention that you may have seen a couple butterflies visit the pawpaw flowers, and someone was asking about the zebra swallowtail, which I do believe is
right.
the pawpaw is the host plant for zebra swallowtail. Would that happen to be the butterfly that you saw visiting the flowers, or what is that connection with the swallowtails? Anything?
It's a great connection. And for my PhD, I studied the whole genus, and down, if you go towards Florida, you get a whole bunch of other pawpaw species growing. There's parviflora and pygmaea and obovata and incana, a bunch of Asimina species. And the zebra swallowtail, the caterpillars will eat pretty much all those, including Asimina triloba, the one up here. I don't really know the range of zebra, so I've seen it a lot more in the south than I do up here. I see other swallowtails around, though, that seem to like pawpaw, as well as a few other species up here, spicebush and stuff like that. The thing is, the swallowtails specifically are looking to lay eggs on leaves. And interesting thing is, all those flowers in pawpaw are usually pretty much done by the time the leaves come out. So the herbivore caterpillar, that's the species that are looking to feed on the leaves, are probably not going to be around as much during pollination time, just because, if their main incentive is to lay eggs on it, they're not there. What we saw was the Polygonia, not sure which species, but the Eastern Kama butterfly, so bright orange, pretty big, charismatic. The times that I've seen it, a couple times, you get ridiculously excited about this because you spend so much time out there with the plants, and there's like nothing visiting the flowers, and suddenly there's this huge butterfly that's just sitting there on a branch, and its proboscis was out, and I didn't mention this during the talk, but the inner side of the inner petals produces this, it looks sweaty, it's nectar, but it's like sweats out of the sides of the petals, and that's what the flies are eating. They're lapping that up while they're getting pollen all over. And this butterfly is just sitting there on, the end of a branch with 3 or 4 flowers around it at different nodes, just probing and probing and probing, and clearly loving this flower and getting something out of it. So both times I saw that, they were feeding. They were really clearly interested. And then I've had friends, because I have a lot of pawpaw friends all up and down the East Coast, mostly, have sent me photos too of other species of butterflies. I haven't seen a swallowtail feeding from the flowers, but kinds of butterflies I don't know that are also hanging out and clearly eating. So, but there are also a lot of butterflies that eat sap, that are sap feeding butterflies. And I think that's probably a really good thing to draw in the sap flows as being potential models for the flowers, because I think the same insects that we go to sap flows are loving the flowers.
Sure. So this one question I feel like might speak a little bit to your comment that you made when you were answering one question about having genetic variability within trees on your property if you were hoping for better fruit set, but I'll just read it to you and you can determine. So If both pawpaw parents in a successful pollination sexual reproduction are from the same clonal colony, how significant is the genetic variability in species throughout an area in which they're growing? Does this have any advantages or disadvantages?
So it's a good question. I haven't looked at genetic diversity within populations of pawpaw, but there are a number of people that have. There was actually a really, really cool study. I think the lead author is Graham, G-H-R-A-M et al., Wyatt Graham. But looking at genetic diversity and population structure for Asimina triloba, they did it broadly over the huge geographic range, but they also talked a bit about this, sort of clonal growth and the impact that has. It's funny because a lot of people will say, well, especially if pollination is, the visitation rate is so low and the fruit set is so low and these trees don't like to pollinate themselves, like, why aren't they extinct? That kind of question. And I just think for a long-lived species that has multiple reproductive events over its life, and for pawpaw, you mentioned being sort of clonal, and if you don't already know this, like the roots send up lots of saplings. So if you have a big mature pawpaw tree, you'll have little thickets under it of just full of pawpaw seedlings, and some of those reach maturity as well. So then you end up with two trees that are both flowering and sexually active with each other, and they're completely genetically identical because they're literally connected underground. I just think because pawpaw is good at clonal growth, that it can persist that way. It can get bigger, it can spread. So sexual reproduction, like actual outcrossing, isn't high on its priority list. And it does just fine making a few really good crops of fruits every now and then and getting the seeds out there. And meanwhile, it's just slow, in terms of reproduction. So I don't know if that totally answers that question, but certainly, these clonal thickets have an impact on the fruit and that kind of stuff.
Yeah, okay. There's one person commented about doing hand pollination with their pawpaw, and there's another down here that also says that we hand pollinate with an artist paintbrush every April and can get 200 plus fruits from true trees. Wow.
That's amazing.
That is amazing.
That's dedication.
Yes. Now they can channel their inner fly when they're doing it this time, right?
Well, I mean, if you want the fruit, that's one way to go about it. And if you have a tree that's happy to pollinate itself too, I mean, that may be enough. It's just, again, there's a lot of variation, and usually a single tree that's pollinating itself won't do too well. But if you were, pollinating with paintbrush across two different genetic individuals, like, good luck.
Yeah.
That should turn out well.
Yeah. I know this is probably not in your wheelhouse, but maybe you've come across it just by talking pawpaws so much. Someone's asking, what other animals might eat the pawpaw fruits once those are produced.
Well, I will say I made it through the entirety of grad school studying pawpaws, Asimina triloba in Columbia, South Carolina, and I never had a fruit, ever. And I didn't spend a lot of time looking for them. I was more out in the springtime than in the fall, but they are so popular. I know possums eat them, squirrels eat them, foxes, I don't know. Those are kind of omnivorous, I think. I don't know foxes all that well, but any mammal that is remotely interested in eating a fruit would eat this. I'm sure in Florida, the gopher tortoises that live down there, and the other species don't make super fleshy fruits, but they eat some of the pawpaw fruits down there and will bury them underground. Yeah, a lot of things like pawpaw. So that's one of the things, finding them in the wild. If you can shake a tree and the ripe ones drop down, get them then, because if they sit on the ground overnight, just one night, they'll have teeth marks in them if they're still there.
I'm just moving into the chat because a couple questions landed there. Just give me 1 second. Do you know about the range of pawpaws? This person is asking how prevalent they are in New Jersey, because this isn't a plant they've really heard of before. Do you know about the range?
Well, I have some good friends in New Jersey who I know love pawpaws. They might even be in the audience right now. So I am pretty sure that at least, I mean, we're talking like central Jersey with a lot of forest, maybe South Jersey a bit. I know that the entire genus Asimina is pretty picky about what its roots are in. So pawpaw likes to be on a slope and near rivers and things like that. It doesn't like to be in super swampy habitats.
Mm.
Although I've seen it in river floodplains and things like that. I just haven't seen it in wet areas. But yeah, I'm pretty sure it does well in Jersey. The closer you get to the ocean along the Atlantic seaboard, you see less of it, but it's in the Chesapeake region all over.
This question is, fly fishermen use fake flies to attract food eaten by fish that they want to catch. Can you use a similar fake fly, a decoy fly, to bring in the right pollinator fly?
Maybe. Flies. It's fun thing with floral mimicry is that flowers that are trying to attract in flies a lot of times put dark splotches on them to look like more flies so that flies think there are flies there. I have never thought of trying to attract flies with fake flies though for pawpaw. So maybe. I bet if you got some, fermented bread dough and put it out and then put fake flies on it, you get a lot of flies. It's a lot of steps to go to though.
Sure. Maybe when you're sitting out there trying to observe flies for hours bored, and you're not seeing anyone, maybe you'll move into the decoy fly.
Right.
Love it. I'm just personally curious if you've looked at any other native plants with floral mimicry. I'm thinking, some of the trilliums and things that also do kind of put out those stinky scents. Have you gone outside of the pawpaw world with mimicry?
I have actually. So just in case you're interested in the future, the Arboretum or anybody, I am now working on skunk cabbage. So it's a super fun thing. Different family, so Araceae now, but really cool plant that heats up. It blooms in like January. It melts snow and burns its way right through frozen ground. And it stinks like carrion. So all those pictures of plants that attract flies because they smell like dead flesh, that's skunk cabbage. And I have now got the whole scent analyzed. There was surprisingly not a published work on the floral scent of skunk cabbage yet. There was a very wonderful study done by a woman in Pennsylvania in 2013 as a master's thesis, but unfortunately with the chemistry involved, they had really identified one compound, which is dimethyl disulfide, which stinks. But the rest of the chemistry was kind of obscure, and the methods that they had didn't analyze it in a way that you could get into those data. So anyway, that's next. That's chapter 2. And that's super great. Same habitat as pawpaw, kind of, just not as swampy. Or it is more swampy than pawpaw.
Right.
Yeah.
Awesome. Well, we'll have to have you back for a skunk cabbage talk sometime. It's such an interesting plant. I mean, the whole thermogenetic part of it. I mean, the whole fact that they can produce their own heat is just amazing.
They're so pretty too. I mean, if you've ever gotten close to skunk cabbage flowers, they're gorgeous. And I think pawpaws are beautiful too. I just love all these flowers. But now I've got like 2 million pictures of skunk cabbage from the past couple of years in my computer, and I'm working on that paper now. Hopefully we'll get it out sometime this year. The scent chemistry of skunk cabbage.
Oh, super cool.
Yeah.
Well, good luck with that.
Thank you.
And thank you so much for being with us tonight. I really appreciate you sharing your time and information with us, and
great questions too from the audience. Thank everybody. That was really great.
And I'm not sure if I had fermentation on the brain when I was scheduling our third Thursday lectures for this fall, but I will say to those of you who are still with us, hopefully you can join us in October for our third Thursday lecture on October 19th. It's going to be on cider, talking about hard ciders and the history behind it. It's called Cider, Pennsylvania's Once and Future Favorite. So we'll kind of dive into the history of ciders and where that's headed. So another pivoting into more fermentation with a different fruit. So
I'll have to be there as an audience member. That sounds awesome.
there we go. All right. Well, Kate, thanks again for being a part of our Third Thursday lectures. And hopefully those of you who are still with us will join us next month. All right.
Thank you, Amy.
Good night, everybody.
Good night.